An enzyme called helicase comes in and unzips the DNA to make room for the 2 new DNA strands to be built. Topoisomerase is an enzyme that keeps the DNA from supercoiling. It binds to the DNA molecule ahead of the replication fork to provide a clear path for helicase. To prevent the 2 strands of the original DNA from coming back together, SSB proteins, or single-stranded binding proteins, bind to them to keep them separated. Then, DNA primase synthesizes RNA primers, or a short RNA molecule on both strands, sending signals to the DNA polymerase of where to start building. Here we can see the adenine being placed next to the thymine and a guanine placed to match the cytosine. Then DNA polymerase builds a new strand in the 5' to 3' direction, meaning it moves along the old template strand in the 3' to 5' direction. On the leading strand, that's right there, you can see DNA polymerase builds it with no problem, but on the lagging strand, primers have to keep being placed in order for DNA polymerase to build. Because the DNA polymerase has to build in the 5' to 3' direction, it keeps having to catch up with helicase, and these fragments are called the Okazaki fragments. So you can see the primer gets placed, DNA polymerase builds, a primer gets placed, DNA polymerase builds, and it keeps going until helicase is finished unzipping. These fragments are called Okazaki fragments. Ligase, then, the gluing enzyme, has to bind together these gaps in between. This continues until you are left with 2 separate DNA molecules. They are semi-conservative because each new DNA has 1 strand of old DNA and 1 strand of newly synthesized DNA.
The process of transcription is copying a DNA segment into RNA. The process consists of 3 parts: initiation, elongation, termination. Before the start of transcription, DNA's double helix has to unwind close to the gene that is getting transcribed, and RNA polymerase helps with the melting of the helix. Initiation. Transcription begins with the binding of an enzyme called RNA polymerase to the DNA strand occurs. This enzyme binds to a specific region on the DNA called the promoter, which acts as a starting point for transcription. The promoter is a specific sequence of DNA that acts as a binding site for the RNA polymerase, as mentioned above, and consists of the tau-tau box. The tau-tau box is a promoter element that is typically found about 25 nitrogenous base pairs upstream of the start site of transcription. Elongation. This is the second step of transcription, where RNA polymerase moves along the DNA strand in 3' to 5' direction and adds nucleotides to the growing 3' end of the RNA molecule. The RNA polymerase reads the genetic information of the DNA, uses the nitrogen base pair ruling, ATGC, to correctly match the nucleotides in the DNA to new RNA. When transcribing DNA to RNA during elongation, uracil replaces thymine. The process of elongation will continue until the RNA polymerase reaches a specific sequence known as a termination signal. The termination signal is a specific sequence of nucleotides that signals the RNA polymerase to stop adding nucleotides to the RNA and to release the newly formed RNA molecule. The copying RNA is called messenger RNA, which carries the gene's protein information coded in DNA.
RNA processing. RNA transcript is first made as a pre-mRNA in a eukaryotic cell, and it must be processed in messenger RNA. The first step is addition of a 5' cap and a 3' poly A tail. The 5' cap is a modified guanine nucleotide added to the 5' end, and the 3' poly A tail is a 50 to 250 adenine nucleotide added to the 3' end. These protect the pre-mRNA from degradation and help ribosomes attach to the 5' end in the cytoplasm. After the cap and tail are added, the non-coding introns are removed from the RNA sequence with the help of spliceosomes. There are several signals in the introns that are used during the splicing process, including the GU nucleotide sequence, the A branch site, and the AG nucleotide sequence. Ribonuclear proteins are in the spliceosomes attached to the ends. The spliceosome binds to the GU sequence, causing it to leave and bend, so the GU sequence is directly above the A branch site. The introns are cleaved to the 5' GU sequence and the 3' AG sequence, forming a last-like structure. The 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the introns are debranched and then degraded. What is left is a matured mRNA with a continuous coding sequence.
The first stage of translation, which is initiation, begins when the small ribosomal subunit gets together with the 5' end of the mRNA inside the nucleus and searches for the start codon, which is AUG. This tRNA carrying methionine, MET, binds to the start codon. After that, the large ribosomal subunit will bind to form the translation complex, completing the initiation. The second stage in translation process is elongation. MET carrying tRNA starts out in the P site. Next, the individual amino acid is brought by the tRNA with the complementary anticodon to the A site. Once they complete the base pairing between the anticodon and codon, the peptide bond is formed, which connects one amino acid to another, and in this case, the methionine, which is attached to the second amino acid at the P site via the peptide bond. The second mRNA shifts one codon to bring the MET codon to the E site, allowing the MET codon to exit the ribosome and exposing the A site for a new codon to enter. Then the whole cycle is repeated until the stop codon is reached. The last stage in this translation process is termination. This stage will happen when a stop codon in mRNA, which in this case is UAA, has entered the inside and no amino acids will be bound after this. The polypeptide chain is then released from the complex, and then the complex disassociates and the next translation process begins.
Control of gene expression for prokaryotes. Prokaryotic gene expression is regulated at the transcriptional level through transcription factors that bind to specific parts of the DNA called promoter regions that either activate or repress transcription. Lac operons is a group of genes that allow for lactose to be used as an energy source and is a positive control. Two regulators, the lac operin and the catalytic activator protein CAP, turn the operon on and off in response to the lactose and glucose levels. CAP sends the glucose and activates transcription of the operon, but only when glucose levels are low. CAP sends the glucose indirectly through the molecule CAMP. The CAP binding site is a positive regulatory site that is bound by the CAP. The lac operon is expressed when the lactose is available and glucose isn't. The lac operon has 3 genes, lac Y, lac Z, lac A, and is transcribed as a single mRNA under one a promoter. The promoter is the binding site for the RNA polymerase at the end of the prevents transcription. The operator is a negative regulatory site bound by the lac repressor protein. Lac Z converts lactose to glucose and galactose. Lac Y codes for permease and is involved in lactosis. Lac A codes for transacinylase. And the lac repressor acts as a lactose sensor, blocking the transcription of the operon, but only acts as a repressor when the lactose is present. The lac repressor can sense lactose indirectly. This is an example of negative control. Control of gene expression for eukaryotes. Eukaryotes don't have operons, so instead, transcription factors and regulatory factors are used. Regulatory factors are made by regulatory genes that could be outside the nucleus, in the cytoplasm, or in the DNA. First, transcription factors allow for the attachment of RNA polymerase to the promoter site. Other transcription factors will hold it in place, but in order to make the gene, the DNA upstream would have to fold. Two, transcription factors bound at the enhancer regulatory DNA sequence on the DNA strand combine to form a dimer and bend the DNA. Once the DNA is folded back, the transcription factors activate the RNA polymerase, allowing it to make RNA and then eventually proteins. Eukaryotes don't have the on and off switches, so the DNA would fold back on itself and activate genes in other places along the genome. This eukaryotic form of gene expression is a more complex form of control.